Assalamualaikum warahmatullahi wabarakatuh. We from the group of 10 will deliver material about metagenomic advance in ecology and biotechnology. Member of our group are Ainaya Salsabila Azara, Esther Felita Wijaya Langodai, and Melinda Setiani Nusa Dinata.
I am Melinda Stiani Sadinata. In here, I will explain about progress of deep ecology, the main metagenomic strategies used to exchange your understanding of microbial ecology and what has been achieved to date. Currently, the field of microbial ecology evolution is growing rapidly with the introduction of phylogenetic diversity studies based on 16S RNA or DNA. This application of direct PCR development of various 16S genes from the total DNA community, combined with the cloning and sequencing, has yielded proven data on low diversity of prokaryotic microorganisms and can the completely refined. Research has shown the microorganisms with identical 16S rDNA sequence have very different whole genomes and display with different physiology and ground temperature optimization, although is except the prokaryotic and physiological diversity go beyond and defined by 16S rDNA or DNA study. Detection and analysis of 16S RNA or DNA remains a very powerful tool in the study microbial ecology, and new information on this variety of software package has been developed by Handelsman Group to help scientists determine whether the difference between the 16S clone libraries to different habitats are ecologically significant. However, RNA, RNA gen analysis still some drawbacks and complex, complex complexity of prokaryotic diversity, and thus of reveal the role different microorganisms is natural population, or it can be cited the 16S gene plays its own role without requiring and role of other microorganisms. One of the main goals of microbial ecology today is the relate the identities of different microorganisms in a habitat to the processes that they carry out in the environment. One way the metagenomic approach achieves this is use 16 as rDNA as phylogenetic anchor to identify clones belonging to a particular microorganism, and when DNA which mark the gene as sequence to obtain information about the physiology of microorganisms. An example of the advance in, in microbial ecology research, the combination of 16S rDNA sequence analysis and metagenome snapshot sequencing approach used to investigation the diversity and process involved high diversity thinking, whether be biofilms. One metagenomic approach that generates large amount of data on microbial ecology is the metagenome reconstruction of ecosystem using a random gene sequencing. It has been used to a successful reconstruct the metagenome from low diversity acidopic biofilms. Analysis of the 16S rDNA clone revealed the presence of only 3 bacteria and 3 archaean lineages. Through metagenome reconstruction, they are able to provide genomic data for the first Lithospora member of the phylum. They are able to allocate process to specific microorganisms and exhibit the metabolic interdependence inter, the interdependence of the biofilm community. An example of challenge in metagenomic research is the identification of clone carrying a specific gene. For example, is 16S rDNA. Among thousands of one of the classic microbial techniques that have been developed in the direct response. One, the need for metagenomic research is fluorescent in hybridization with the use of a fluorescent probe to detect 16S RNA. The combination gene expression or activity screening with phylogenetic screening for 16S RNA using the large insert library fish has proven successful. The possibility that this could be combined with fluorescence activated cell sorting would provide a useful technique for phylogenetic screening of large metagonic libraries. The main drawback of this method is the cells must be filled with paraformaldehyde prior to hybridization with an oligonucleotide probe, which effectively kills the cell and prevents the isolation of phosphate DNA after cell sorting. PCR sending may be a suitable approach for obtaining phosphate DNA after cell fixation.
The ability of prokaryotes to adapt, develop, and inhabit any environment, from hydrothermal vents on the ocean floor to acid mine drainage sites, is related to their metabolic and physiological diversity. Okay, next, function-based screening for the biokinetic pathway, which consists of type 1 polypeptide synthase involved in the production of antibiotics such as erythromycin, rapamycin, or epinephrine, use both E. coli and as evidence as heterologous hosts and result in detection of H-positive clones from 5,000 metagenomic libraries. Soil has always been one of the main sources of antibiotics, although the number of new antibiotics detected by culture-based techniques has surged in recent years. Analysis of the metagenomic library derived from soil has revealed that there are more antibiotics present in the non-cultivated population. Okay, next,
A new antibiotic, palmitofluoxetine, was discovered in bromelain tank water and a novel bacterial mechanism of tetracycline inactivation was discovered in the oral metagenome. One of the main benefits of applying metagenomics in this area of researching is increasing knowledge of the mechanisms of bacterial antibiotic resistance and enabling the development of compounds that inhibit resistance effectively, expand the functional range of antibiotics. Cultivation is usually avoided in metagenome studies because of loss of genetic diversity and introduction bias in results. Pre-cultivation can be a useful tool for obtaining sufficient DNA from the soil environment and can be offset by loss of diversity, the significance of which depends on the natural level of diversity among microorganisms carrying the required properties. One of the main areas of metagenomic research from its inception in its application in the discovery of new biocatalysts. Given the high prokaryote biodiversity in the cell environment, the cell metagenomic library offers one of the best sources when looking for various biocatalysts. However, often thousands of clones must be analyzed before even a small number of positive clones can be detected. One of the developments that has allowed the rapid screening of a large number of clones is the use of DNA microarrays. Using this approach, it was possible to identify clonal products from non-cultured microorganisms, thereby narrowing the range of clones to be sequenced and analyzed further. A similar development of microarrays, very sensitive at the protein level, allows detection of animated activity in hundreds of samples simultaneously.
I am Ainaya Salsabila Azara will present the conclusion of ecological progress and biotechnology progress. Ecological progress. The field of microbial ecology is revolution with the introduction of phylogenetic diversity studies based on 16 RNA or DNA. The application of universal primers for direct PCR amplification of various 16 genes from the total DNA community combined with cloning and sequencing has resulted in a large amount of data and a complete redefined prokaryotic diversity. Soils which were shown to contain low diversity of microorganisms when the analysis using culture-based technique are now, as a result of direct analysis of the 16 RNA gene, which is recognized as one of the most richly diverse habitats for prokaryotic life on Earth. Recently, various software packages have been developed by the Handelsman Group to allow scientists to determine whether the difference between the 16 clone library of different habitats are ecologically significant. From the ground, this approach has been extended to include other Archaea-specific genes as well as 16 DNA for identification of clones containing DNA from Archaea. This effectively expands the amount of genomic information that can be accessed and provide insight into process at play. Finally, they were able to demonstrate the many different special abilities this biofilm exhibits for survival in highly acidic and highly toxic environment. Biotechnology progress, the ability of prokaryotes to adapt, develop, and inhabit any environment, from hydrothermal vents on the ocean floor to acid mine drainage sheets, is related to their metabolic and physiological diversity. The metagenomic approach offers the possibility not only to analyze the phylogenetic diversity of environmental biofilms, but also to find genes and operands encoding properties of biotechnological interest. Using the Ks domains of the clones and comparing them with the resulting phylogenetic trees of 23 known Ks domains, it was possible to find clones containing the new type, polygated biosynthetic pathway, and thus prove that screening soil metagenomic libraries is a valid approach for detecting new biosynthetic pathway for product of therapeutic interest. One area of new drug discovery made possible by metagenomics is the investigation of symbiotic bacteria, one of the main natural sources of therapeutic products and which have so far resisted analysis by pure culture technique. One of the main benefits of applying metagenomics in this area of research is increasing knowledge of the mechanism of bacterial antibiotic resistance and enabling the development of compounds that can inhibit resistance, effectively expand the functional range of antibiotics. A variety of different biosynthetic pathway accessed by metagenomic studies are involved in vitamin biosynthesis. If there are questions, you can ask our group. That's all from our group. Wassalamualaikum warahmatullahi wabarakatuh.